Hello, lifelong curious people! Today we're going to talk about ways to package small payloads for delivery into and out of your cells. Luckily, our bodies are already pros at this. Vesicles are tiny packets in your cells that have stuff inside, commonly referred to as the payload. They're generally 30 to 100 nanometers in size. Nanometers is also a fine way to pronounce it. In fact, I often switch back and forth, so if I switch back and forth in this video, please bear with me. Being so small lets them fuse easily with other membranes, be it cellular membrane or an organelle's membrane. Being on the nanometer scale means that they are considered a nanoparticle. For example, neurotransmitters inside the cell need to be at the ready for when a synapse needs to be activated. But before the synapse needs its signal, we don't want it activating anything else. To avoid this, the cell will keep it in a nice little package made up of the same material as the cell membrane. When the cell finally wants to shoot the signal out towards the other cell, the package will actually fuse back into the cell's membrane and become part of the cell's membrane. Just like this. Thank you to Quicksilver Scientific for use of this excellent animation. I'll link the full video that this animation is from in the description. Quicksilver uses a liposomal delivery system to bring their supplements into people's cells. I'll be talking about liposomes specifically a little bit later, but they work nearly identically to vesicles because they're basically vesicles themselves. Just a heads up that this is not a sponsored video, but I do work for Quicksilver. Also, anything I say in these videos are my own thoughts and my own research. It does not reflect on the company or how the company thinks or talks about these things. So again, these vesicles will actually fuse themselves to the membrane, becoming part of the membrane, and shooting their payload to the other side of the membrane from where they started. Once the payload is in or out, as the case may be, it can do whatever it's supposed to do, like finding a cell with the right receptor or doing enzymatic work. For our example, since the cell is secreting the payload that was inside the vesicle, that vesicle is called a secretory vesicle. Biology is very inventive with the names, I know. Medical News Today groups vesicles into 5 categories. Transport vesicles. Cells are incredibly goopy. This insanely high viscosity means that if you want to reliably get something from one side of the cell to the other, then you gotta package it up and carry it there manually. Even once it's in the vesicle, the whole package needs to be towed across the cell by a kinesin. Yeah, that little guy. Lysosomes. They digest things. In macrophages, the immune cells that eat bad or dead things, they'll make a vesicle to eat the bad stuff without letting the bad stuff actually touch their cytoplasm. And then the lysosome will fuse with the garbage bag membrane in much the same way that the secretory vesicle fused with the outer membrane. This shoots the incredibly toxic substances that the lysosome was holding into the garbage bag, dissolving and rendering harmless whatever was in the garbage bag. So they put peroxisomes in a separate category to lysosomes, but I really think of them as a subcategory of lysosomes. They'll break down lipids and peroxides, sure, but I feel like the main reason that they're their own category is because they also work with liquor. So either way, they also break down things in the cell to render them harmless. If you know why they're their own thing, please let me know in the comments. Secretory. We talked about these first, but these are the vesicles that shoot stuff out of the cell. There's a variety of stuff that could be, but think sending signals across a synapse by shooting the neurotransmitters to the cell on the other side. And extracellular. Extracellular vesicles aren't extremely well studied, but it looks like they do intercellular or cell-to-cell communication. Since these little cellular packages are such a great way to get things into cells, people have used that concept to make their own for drug and supplement delivery. When I started researching this video, I knew that there were several ways to deliver drugs via nanoparticles, but there are so many ways to do nanoparticle drug delivery. We'll start with the one I'm most familiar with, liposomes. Liposomes are basically doing the same thing that your body does. They're made out of phospholipids, like the membrane is, and thus will readily fuse to any membrane if it's small enough. If it's bigger, then the cell won't want to absorb it. This is a good thing, or else you'd have cells absorbing other cells all the time. We don't want that. We like being multicellular. So people package their payloads in as small of a liposome as possible. When you're getting under about 250 nanometers, the cell will start having okay uptake. But generally, the smaller you can get it, the better it'll be uptaken. This picture is also supplied by Quicksilver Scientific, showing what it looks like when you get them really small. If it looks a little cloudy, like that one, it's gonna be in that range. That one is much less cloudy, so it won't be surprising to find that it's slightly smaller in that range. The last one is basically perfectly transparent, so a trained eye will know that it's under that many. If you get a product that claims to be liposomal, but then you look at it and it's super cloudy, run for the hills, because it may be liposomal, but that won't be absorbed by your body. This is a well-used and well-loved delivery system at this point. Liposomes are also the delivery method for the mRNA COVID-19 vaccines. All the articles talking about the vaccines call them lipid nanoparticles, but that's just another word for liposome. Degradation of liposomes has the extra happy benefit of replenishing the cell membrane. If you make the liposome out of phospholipids with certain properties, like having mostly unsaturated fats in their tails, you can even increase your membrane's fluidity, which helps facilitate other functions, like getting receptors where they need to be more easily. I had a lot of fun researching the rest of these for this video. Much of the information can be found on the Wikipedia page for nanoparticle drug delivery, linked in the description, and that's also how I grouped my video. But I also hit up Science Direct and a few other sites to fill in and deobfuscate the information from the wiki. I'll link to some of the sites that I used for information below if you want to peruse. Okay, so polymeric nanoparticles are the overarching category that includes silicon dioxide nanoparticles, which is what's used in the article that I made this background video for originally. Polymer just is referring to repeating units, so this category encompasses a whole bunch of levels of toxicity and uptakeability. Ones like silicon dioxide are easily modifiable and easily broken down in the body to either be used by the body or excreted by the kidneys. Size versus toxicity varies depending on what it's made of and how it's made. But they'll carry whatever payload you want and will fuse to more specific cells if you modify them to activate certain receptors. Inorganic nanocrystals are usually made up of an inert metal like platinum, and they can get really extra small. But they will always need to be functionalized, which just means that they'll have to stick something on it that'll interact with whatever they want it to interact with. Depending on the molecule that they use to functionalize it, it'll be absorbed or link up to a receptor or look good for being eaten or whatever they want it to do. With these guys, you'll want to be careful about toxicity and bioaccumulation. If you're using a metal that isn't used by the body very much, which you often are, then it will take a while for it to actually be excreted out of your body. Organic nanocrystals are literally just making crystallized forms of the payload. It takes a very specific payload to make this feasible. So be careful if choosing this method if you're trying to get it working all the way into the human. You'll need to look at the drug's stability and how hydrophilic it is, as well as making sure it won't wreak havoc if it's released all at once. If it's toxic in high concentrations, then you're going to want to be very certain that it's not going to dissolve immediately once swallowed. Another fun way that people have figured out how to get things all the way into the nucleus, so things like genes to be integrated into the DNA, is by using a virus. This little guy is called a bacteriophage, so he is more suited for getting into a bacterial cell, but he's similar enough to viruses that infect us that he'll be a good prop. If you take this protein coat and take the DNA or RNA out of this little box here, you can put in your payload, including whatever viral particles will actually help integrate the DNA into the target. Humans are freaking ingenious, am I right? Okie dokie, so that is a crash course in nanoparticles, inside the body and out. Our body uses vesicles for a whole bunch of things. Packaging payloads to be moved, digested, ingested, or excreted. Humans looked at that and thought that was pretty cool, so we make our own nanoparticles with various properties that will deliver our payloads. If you're looking into using them, you'll want to think about whether you're trying to target specific cells, about bioaccumulation, and how toxic it can be. Please let me know in the comments if you have any questions or insights into the world of nanoparticles. Join us on Instagram at Academic Article Book Club and on Twitter at Academic Article 2. I wish you exciting new discoveries. May your day be wonder-filled.
Not yet recording, so recall. Yeah.
It's on and recording. For example, neurotransmitters. For example, neurotransmitters inside the cell need to be at the ready when a synapse needs to be activated.